Several steps have to happen before a muscle can contract. These steps can be divided into kind of 4 main steps, and then we'll talk about each one in more detail after we introduce them here. So the first thing that has to happen is excitation. So in order for a skeletal muscle fiber to contract, it has to be stimulated by something. And it's typically stimulated by a special type of neuron called a motor neuron. During excitation-contraction coupling, the stimulation from the neuron spreads across the sarcolemma, which is the cell membrane of the skeletal muscle fiber. This ultimately causes the sarcoplasmic reticulum deep inside the cell to release calcium. That calcium is then involved in making the actual contraction happen. So the contraction is when the myofilaments of actin and myosin slide across each other, and this produces the shortening of the cell, which gives you that contraction and pulls 2 different structures closer towards each other. And then after the contraction happens, everything that happened in the first 3 steps has to stop and be reversed in order for the muscle to relax. So let's first take a look at excitation. So in excitation, a neuron stimulates a muscle fiber. We took a look a little bit at neuron structures back when we talked about histology and tissues. So we can see that this is kind of a cartoony drawing of what a neuron looks like. And you've got the head of the neuron, I'm sorry, not the head, we call it the cell body. And the cell body has these dendrites attached to it. And then there's one long cellular extension called an axon. And then that axon at the very end branches into axon terminals. And these axon terminals form synapses. And a synapse is just anywhere where part of a neuron meets up with something else. So down in these axon terminals, there are, you can kind of see it here actually. So these are muscle fibers, these long kind of red cylinders. And then these are the axon terminals. And inside the axon terminals, you have these little bubbles or structures called vesicles. And these vesicles contain neurotransmitters. And the neurotransmitter is how the signal from the neuron travels to the skeletal muscle fiber itself. So now that we know what a neuron looks like, let's take a look at the actual steps involved in that excitation. So the neuron itself does something called a nerve impulse, which is also called an action potential. So what happens is the action potential travels from the cell body and the dendrites down through the axon to the axon terminals. When the action potential reaches the axon terminals, it causes these little bubbles, these vesicles, to release its neurotransmitter. And in the case of a skeletal muscle, the neurotransmitter is called acetylcholine, which we can also just abbreviate as ACH. So what happens is that acetylcholine gets released from the axon terminal and actually goes into a little space in between the cell membrane of the neuron and the cell membrane of the muscle fiber. So this is the sarcolemma or the cell membrane of the muscle fiber here. So this space in between is called the synaptic cleft. So that acetylcholine neurotransmitter is released by the axon terminal into the synaptic cleft. Once it's in the synaptic cleft, that acetylcholine binds to these little purple receptors here. And these purple receptors are embedded in the cell membrane of the muscle fiber. When that acetylcholine binds to the protein, it opens it up. And these proteins that the receptors are bound to are sodium channels. And so what happens is the acetylcholine, the neurotransmitter, which is this pink dot here, binds to a receptor on the sodium channels. When that neurotransmitter binds, it opens up the sodium channel, and sodium diffuses from the synaptic cleft into the sarcoplasm sarcoplasm of the skeletal muscle cell. Now, as that sodium moves into these little channels here, it actually causes a domino effect where additional sodium channels open along the rest of the sarcolemma. And remember the T-tubules, so those things that were basically those indentations of the cell membrane going deep down into the cell, that information or that stimulus travels down those T-tubules. So once again, to recap, we open up sodium channels here with our neurotransmitter. You can think of the neurotransmitter as kind of like a key, and the sodium channels are locked until the key unlocks them. But once they're unlocked, they open up, and sodium diffuses from the synaptic cleft into the sarcoplasm. Once these sodium channels open, sodium channels over here are also going to open, and sodium channels in the T-tubule are also going to open. So it's a domino effect where these are opened by acetylcholine. The ones that would be out here are opened by the fact that the other sodium channels are already opened. So once you get it started, it kind of goes from there. Now during excitation-contraction coupling, and we're just picking up where we left off, so remember the stimulus was traveling along the sarcolemma or the cell membrane down into the T-tubules. Now remember, on either side of the T-tubule you have the endoplasmic reticulum. So as that stimulus travels down the T-tubule, it stimulates the sarcoplasmic reticulum to release calcium into the sarcoplasm. So this structure, which is this structure here, this is the sarcoplasmic reticulum, stores calcium. But when the signal from the sarcolemma travels down into the T-tubules, it triggers the sarcoplasmic reticulum to release calcium. That calcium goes into the sarcoplasm and it binds to troponin. Remember, troponin is one of the regulatory proteins that is attached to tropomyosin. When calcium binds to troponin, it pulls on tropomyosin and it pulls it away from the binding sites on actin. So the little dark circles here are the binding sites. So those have to be open in order for myosin to be able to grab onto actin. So as long as there's calcium present, the tropomyosin is moved out of the way and the myosin can now bind to the actin. So the myosin head, after it binds to the actin, the head itself can bend. And when it bends, it pulls this actin molecule along the surface. Now, this this diagram here is just showing one myosin head, but in reality there would be a lot in this area. And so typically the myosin head will bind to an actin site and pull on actin, and then it can unbind and rebind at a different spot and pull some more. So each bend of the myosin head only pulls the actin a really small amount, but then it can unbind and pull it again. So it's kind of like when you're pulling a rope with two hands. As one hand is pulling, the other one can be releasing to grab the rope further up to pull it even further. Now you'll notice that ATP is required for this process to happen. So your skeletal muscles require a lot of energy in the form of ATP in order for the myosin to be able to walk along actin. Now remember that myosin and actin are arranged in these structures called sarcomeres. So we've got our Z discs here, we've got our actin here, and our myosin here. So as these little myosin heads pull on actin, it pulls the actin, it pulls the two Z discs towards each other. So here we can see a relaxed sarcomere, and here we can see a contracted one. So you see how the Z discs have moved a little bit closer together, and the actin molecules actually meet up with each other. So there's a little bit of a gap here between this actin and this actin, but down here you can see that this actin molecule actually meets up with this actin molecule. So notice that the length of myosin does not change, and the length of actin does not change. The thing that gets shorter is the sarcomere, and it's just because the Z discs, so the sarcomere is this area, and the Z discs are moving closer together. So when a muscle is relaxed, the sarcomeres are longer. When it contracts, they're shorter, and that's because the myosin pulls on the actin. Now, each myosin head can only pull a short distance, because remember, we're looking at, this is a whole muscle fiber here, and then this is just one myofibril with one sarcomere here. So remember, we're looking at this underneath the microscope. So this is very, very small. So each myosin head can only pull on actin a very short distance. But each myofibril, so each one of these structures right here, has thousands of sarcomeres. So thousands of these units right here make up each myofibril, so one of these. And then each muscle fiber, depending on its each muscle fiber, depending on its size, may have hundreds or thousands of these myofibrils. And then each muscle fiber has about 1 million sarcomeres, and that's in one cell. So depending on the size of the muscle, it can be made up of a lot of cells. So we're talking about potentially billions of sarcomeres that are all working together to accomplish the contraction of a single muscle. All right, so sarcomeres are very, very small, but you have a lot of them that make up the overall muscle, and they work together to give you that contraction. Now, after a muscle contracts, that muscle needs to be able to relax. And so relaxation just involves all of the steps of excitation, excitation-contraction coupling, and contraction. All of those events have to stop and be reversed. All right, so if we go back to the very beginning, the neuron is going to stop sending the stimulus. The acetylcholine is going to stop being released by the neuron. And that acetylcholine actually, the acetylcholine that's kind of left in the synaptic cleft gets broken down by an enzyme called acetylcholine esterase. So without the acetylcholine present, those sodium channels in the sarcolemma of the muscle fiber are going to close, which means that sodium stops entering the cell. Once that sodium stops entering the cell, the action potential along the cell membrane of the muscle fiber also stops happening. Without that action potential stimulating the sarcoplasmic reticulum, the calcium channels are going to close, and the sarcoplasmic reticulum is no longer going to release calcium. And in fact, it actually transports that calcium back into the sarcoplasmic reticulum in order to be used for a future contraction. So the calcium then, it's unbinding from the troponin. Without calcium bonded to troponin, tropomyosin shifts back in place to cover the myosin binding sites on actin. So if myosin can't bind to actin, those sarcomeres are going to slide back to their resting length, and the muscle will overall change from being contracted or shorter to being longer and to its resting lengths.